Dear students, I hope you're all well and studying in your home. Last week we discussed about seed and its types and we compare a dicot seed and a monocot seed with their images. So what com- uh, comparison we show last week between bean seed and maize grain. I hope you read also so we can conclude this comparison in a table. So first is seed coat is thin and brownish in color in bean seed, and seed coat is very thin and fused with pericarp in maize grain. And helium and micropyle are visible in bean seed, but helium and micropyle are invisible in maize grain. Two thick, fleshy cotyledons are present. Single thin cotyledon present because it is a monocotyledon, as you know. Endosperm absent. It means it's non-endospermic bean seed, but it is endospermic maize grain seed. Reserve food is carbohydrate and protein is present in the cotyledons. Reserve food material is starch present in endosperm. As you know, in monocots mainly endosperm is found and it provides nourishment to the developing embryo. Tips of plumule and radicals are naked. Tips of plumule and radical are covered by protective sheaths called coleoptile and coleorhiza, respectively. And difference between dicot and monocot seed. So monocot seed, the seed contains single cotyledon, as we know, and in dicot, it possesses two cotyledons. The food is commonly stored inside the endosperm. Okay, one exception is there here, orchids, okay, in which not stored. The food may be stored inside the endosperm or cotyledons. An allurine layer of a special protein rich cells is present outside the endosperm. The layer is absent. The embryo tips may bear special seeds, coleoptile over plumule and coleoza and over radical in monocots. In dicots, these are absent. Embryo occupies one side of the seed, and embryo occupies the whole interior or only the central part of the seed in case of dicots. In monocots, plumule lies at one end near the cotyledon, and plumule lies in between the two cotyledons. Now we start our new topic of this chapter, seed dormancy and germination. In most cases, the seeds are dry and incapable of germination when set from the parent plant. In this condition, all the metabolic activities are taking place, but at the lowest rate just necessary for survival. You can say that in unfavorable conditions, okay, the seed goes to dormancy. A very small quantity of stored food is used. The seed which do not germinate even when provided with all suitable conditions necessary for germination are called dormant seed. This phenomenon is termed as dormancy. Okay, it is very important in overcoming the unfavorable condition. It helps in dispersal as well as storage of seeds. So now we talk about germination. The sum total of all processes by which dormant embryo of the seed grows out of the seed coat and establishes itself as an independent seedling is called germination. Okay, when suitable conditions are available and the embryo has overcome dormancy, it becomes active and it starts germinating. The process of germination is irreversible. Okay, it means once seed is germinate, it cannot be goes back to its dormant stage. Okay. There are some conditions necessary for the germination of seed. First is water, availability of O2 and moderate temperature. So some seed you can easily sow in your home also. Okay. When you soak some gram in water, it will be eventually you can see that it eventually swollen after one night, okay, and or after some time in summer season. So and if you remain it for many times, it will be start germinating. So dry seeds contain only 10% to 20% water. They are incapable of germination as long as sufficient quantity of water is not available. So when their germination start only when they gives with water through the micropyle and absorption through their surfaces. Okay. Next is oxygen. During germination of seed, metabolic activities accelerate and require energy. This energy is released in from the stored food material by the process of oxidation. For this purpose, the seed require oxygen. Because of oxidation of food, oxygen is consumed and CO2 is produced. This exchange of O2 and CO2 takes place primarily through the micropyle. Temperature play an important role. Okay, so seed of different plants germinate at optimum temperatures. Temperature at which seed grow most rapidly. Generally, seed of tropical plants need higher temperature, whereas that of temperature climate need low temperature for germination. Process of seed germination. The seed contains a dormant embryo. When it is to germinate, rapid intake of water through the micropyle makes the seed large, swollen, and the seed coat becomes soft and burst open. Absorption of water induces a number of physiological changes in the seed and enhance the respiratory activity. The embryo produces a large number of enzymes. These enzymes hydrolyze the stored food material and make it available to the growing embryo. With the availability of food, cell division start in the embryo, radicle, and plumule. Grow in size and emerges out of the seed coat. Thus, radicle is first to come out of the seed coat. It forms the root system that grows into the soil. The plumule, after coming out of that seed coat, forms shoot system. Okay, mainly I told you many times that the function of this uh, plumule and radicle. As mentioned above, the germination of seeds requires sufficient quantity of water, air, and outdoor temperature. It can be provided with following experiments. Okay, so some experiments also there that to demonstrate the water is essential for germination. Okay, to demonstrate oxygen is necessary, or to demonstrate temperature. Okay, what is the effect of temperature? So for this experiment, bean seed soaked and unsoaked, 4 patty dishes with lids, cotton wool or blotting paper, etc. I hope you can do this activity with gram seeds also, and you already do in your previous classes. Label the 4 patty dishes as A, B, C, and D. Place the cotton wool or blotting paper and seed at the bottom of patty dish as follows. Okay. In Patrice A, the cotton wool or blotting paper at the bottom should be moist. Okay. In Patrice B, seeds are soaked in water but are placed on the cotton wool or blotting paper which is not moistened. Unsoaked seeds are placed on the dry cotton wool. And next, soaked seeds are completely submerged in water. Like this, you have to do first A Patrice, B Patrice, okay, C and D. Then you have to observe. Okay. After keeping the above petri dishes at room temperature for 2 to 3 days, the following observations are made. Seeds germinate properly in petri dish A. Seeds may begin germination but shrivel up and die in petri dish B. In petri dish C, no change is seen in the seed. In petri dish D, the seeds may start germinated but eventually rotted. Okay, so what will be inference from this experiment? Proper germination of seed needs adequate quantity of water as in petri dish A. In the absence of water, seed do not germinate as in petri dish B. Deficient and excessive water also does not allow the germination to continue because O2 is not available. Once the O2 dissolved in water is used by germinating seeds. Okay, there are some precautions in this experiment also. Each dish must contain equal number of seeds from the same source. All the 4 petri dishes must be subjected to similar conditions of light and temperature for the duration of the experiment. Okay, the petri dishes should be kept covered with lids to prevent evaporation of water. So you can see that in this experiment you have to do uh, adequate quantity of seeds, okay, and petri dishes are of equal sizes, etc., and optimum temperature and water will be provided, and you have to do experiment like this. Okay, you can also do this experiment. Now you can read experiment 2, the effect of temperature on germination of seeds. Okay, or we can discuss it with the next class also. Okay, or if you read this, it's very good, because these all are three experiments that to prove oxygen is essential for third experiment also here. So meet you in the next class. Thank you.